Well, good morning, everyone. Welcome and greetings. This is an exciting opportunity for us with the KIBM symposium this morning and um, the Institute for Neural Computation retreat in the afternoon. KIBM has been partnering with INC for a number of years now. We enjoy these meetings tremendously. Of course, this is the first time we've been doing it as a Zoom, but many of us have been doing a lot of Zooming. So I think that this should go very well. And Megan Ensminger is helping us out with a lot of the logistics. So I think she and others really got us up and running here. This is the 15th year of awarding innovative research grants. It's quite a milestone. I want to offer congratulations to this year's awardees. There were 10 of them. You can find them on the KIBM website. And tough to make these awards. There were 40 proposals. We'd have been delighted to fund another 10 or more, but there's only so much money to go around. So we're of course working hard to try to increase the endowment, but these were great proposals and we're very happy about that. I want to take a quick moment here to recognize the incoming co-directors of KIBM. Rusty and I will be stepping down in a few days, and Yishi Jin from UCSD and Ed Calloway from Salk will be stepping in, and we're very excited about that. This is going to be terrific. In addition, I want to recognize the two associate directors, Ralph Greenspan continuing from years past, and importantly, Gabe Silva from the Jacob School will be joining us. And we think this is wonderful because we've seen a growing connection to the Jacob School and engineering, and this really strengthens that relationship. So this morning in the symposium, the KIBM symposium, we'll hear from 8 of the last year's awardees. Exciting science. Again, as you know, we're trying to bridge brain and mind here, reaching out to bring these two camps that had in the past been somewhat separate, trying to integrate them. And I think this has been very rewarding. The format, as you know, involves a 10-minute talk and then 5 minutes for Q&A. And please type your questions into the chat. There's a little icon at the bottom. If you're not familiar with this, you just click on that and then a screen appears on the right, type it in. And then I'll be looking at that, and we'll hope that the speakers will leave some time for us to do the questions afterward. So I think with those formalities out of the way, let me bring forward Gerald Powell from Salk Institute. And Gerald's presentation is entitled Downloading Brain Activity into Low-Dimensional Manifolds. So Gerald, please share your screen.
Yes. Thank you so much for having me over this morning and giving me this opportunity to present the work I've done with my Kavli Award. And yeah, today I'll talk about downloading brain activity to low-dimensional manifolds. And I, just to state what the goals I had going into this, it sounds a little bit, lofty, but it's like, basically I wanted to develop methodology to basically take brain activity of an organism that's recorded and then put it into a computational form that can reproduce both the neural activity as well as the behavioral outputs in a way that inform each other. So it's basically, if you were to run an organism in a computer in terms of the neural activity and the behavior, would you be able to do that or to put it into a robot? That was the sort of the goal. So the general mathematical framework by which I do this is by using these things called low-dimensional manifolds. And manifolds are basically shapes in this artificial space. This is called phase space, where you actually have the starting material is basically time series. So a time series is like a time-varying signal that you have, and you were to plot this against each other, like several time series, you can have like here, X, Y, and Z of the Lorenz attractor. If you were plot every point against each other, what you generate is the shape which defines the relationship between these three time series. And this is arbitrary, whatever you use. But if you actually have some relationship, this relationship will turn into this shape that we call manifold. And you can go from a manifold to the time series, and you can go from a time series to manifold. So let's say we take the neural activity as time series of neurons that have some relationship with each other, and then you will start making things. One important thing here is that, in 1981, there was this thing called the Takens theorem was developed. And the Takens theorem says that even if you don't have all the variables, so even if you don't have all the variables, if you have one of the variables, you can actually generate a shape that has basically, it's a stretched form of the original manifold. So for example, in this thing that you see here, this is like just using the variable x, you can generate this manifold, which is a stretched form of the original one that had the variables x, y, and z. And this is just the basis of generating a whole set of tools. I'm not going to explain them in detail, but I'm just going to let you know, because we don't have enough time, to let you know what you can actually do with these kind of things. And one of the things that you can do is you can predict. You can use these things to predict. Let's say you have a manifold. I'm in this position. Where is it going to go next? I can predict what's going to do next. So I can predict what's going to happen to the time series. And then you can also, in 2012, George Sugihara does down at SIO, developed this method that allows you to do causal inference. It allows you to find the cause and effect relationship between things that are connected, but they may not be. And then, as for example, you can actually have, like I said, this is the original system. You can generate a manifold for the variable x and generate a manifold for the for the variable y. And then if you can see that you can predict using x, predict something about y, they share information, and then you can infer basically that they're causally related to each other. And yeah, that was basically the basis of that 2012 paper. So using some of these techniques and some others, in addition, what we did is we took the brain activity of one of the papers of Terry and Ralph Greenspan that was done by Sophie Aymon, who used to be here with us, and took the brain activity of using light field microscopy of GCAM activity, and generated 80 areas through PCA and ICA. So you generate basically 80 time series. And you also have the movement of the fly on a styrofoam ball. So you can generate those time series and then find the causal relationships between these things. And what we did is we find the causal relationships, and then we develop a new technique to generate manifolds out of the neural activity. Something like this. This is a neural activity out of one neuron. But we actually generate from all of them, and we're going to want to generate manifolds for each of these neurons. And then we take the CCM predictions, and then we subtract the correlation from these and look at the nonlinear residuals. And these residuals tells you basically what is the non-redundant information. And using this non-redundant information, we can build manifolds out of this. And this is basically what we use is that we generate a row diff matrix, what we call, which is basically the CCM prediction skill minus the absolute of the correlation. So how do you make out of these things a manifold? So basically you just take a bunch of time series that, using the causal inference stuff, you can know they can predict each other a little bit. So basically this is basically using this variable like time series 49, which corresponds to a particular brain area, you can predict the forward movement with this skill. So that's the row. So basically 39, the cross correlation coefficient of the predicted values from the observed values is 0.39. So you can set some prediction about background, but it's not terribly good. But you actually put it all together and you generate a manifold out of these time series to predict forward. But what we want to generate is a network of manifolds. So what do you mean by that? So let's say you want to predict left and right movement. You have the time series of left and right speeds. And then you actually have two unknowns. So you have to solve this thing that you only have one unknown. So what we actually do is basically we make a time series that gives you that predicts the forward. And then we take the instantaneous values of the left, right, plus the predicted forward. So we eliminate the two unknowns to a single unknown. One is from the prediction, and the other one is other two time series that independently were able to predict left, right. And then we added the predictions from the first manifold. And since there are also other neurons or neural areas, you actually build up equivalent manifolds that predict each other, and then you generate a network out of it. So the network that you actually generate is, this is basically out of the 80 time series, 64 of them were used, and then we can actually see like which which neural areas predict which other ones, and how do they integrate information. And ultimately, they predict this behavior, which is left, right speed, and forward speed that you see up there. And this entire network just is from the inferred causal relationships. So typically, when you actually do these manifolds and basically this is the training set on the blue, and then you start predicting things in the future, these things kind of collapse, and they get into these local sinks. But when we actually use a network, these actually produce fairly real. So this is basically forward movement. And when we try to use this predict forward movement, it produced fairly realistic looking kind of time series of this forward movement. And we can also do this for neural activity. So this is the training set that we use to generate this. And then you can actually generate like pretty realistic looking, artificial data of neural activity. So how similar is this? We want to have some metric. So we did the Fourier transform of the basically of the forward movement, or we can do it for every single neuron and every single movement. You can see they look fairly good. It's like you can have a correlation coefficient of 0.92 of observers is expected. So it has some semblance of this thing. If you actually look at the real thing, you can see this is the original movements. This is the forward speed, and this is the left-right speeds of the fly on the styrofoam ball. And this is what you actually generate from the downloaded brain activity that we generate. But if you actually, so just to exclude the possibility that we actually might be getting it from the locomotion itself, if you just use the locomotion data, you cannot do it. What is interesting also that we had this training set, there was withheld data. We only used the first 600 time points. In the generated activity, you actually see these pauses that actually exist in the withheld real data that were not present in the training set. So something in the network knows how to do this when you actually don't have them. So there are some emergent, and you can actually see, so then we want to compare it to other types of generated. So if you use the left-right movement alone, they actually generate these forever periodicities. Or if you use a flattened network, where you make a manifold of all 80 time series and try to take all the information and put it into a single manifold with 80 dimensions that generates, you get these forever loops, basically, these repetitive movements that keep going forever. So here we actually compare the true data with the generative manifold network, left-right movement alone, what we call flattened network, is 80 time series embedded into one manifold, basically an autoregressive model, and the k nearest neighbors, and in all cases, it performed much better. So the ultimate test that we wanted to have is basically, what can you do to see if these time series are really realistic? So we actually generated artificial data from the model, and then see, can we actually use this to predict the real data? And you can see that we can actually do this fairly well. And this is going from 5 time steps ahead up to 30 time steps ahead. Whereas like if you use left-right movement alone or flattened network alone, it degrades very well. And MAE is the mean absolute error. So you can see the real data is the blue one, the predicted one is the orange one. And here we actually have basically a measurement of the prediction skill across with the different things. You can actually, this is for every single neuron, neural area from the fly brain, but it actually is better than you think, because if you actually look for every single area, in almost every case, this what we call generative manifold network can actually generate data that can predict neural activity, the real withheld neural activity, better than any other model. What is more is like it can predict, this is just a validation scheme. So anyway, the conclusion is that here we actually have generated a method that can generate an embedded representation of brainwave activity. It can produce realistic walking behaviors from neural activity. It can generate non-repeating realistic brain neural activity. And also it can generate things that are not strictly present in the training set. So it has emergent properties, and it's have something about it that allows you to have sort of like a realistic portrayal of brain activity. So anyway, so that is pretty much it. And this was completely funded by the Kavli. And this was just done by myself and an undergraduate student, Cameron Smith, in computer science at UCSD. Okay, thank you very much.
Thank you, Gerald. That was fascinating. We have some time for questions. Let me jump off with one of them. What are the prospects for generating larger manifolds to try to simulate and replicate and predict more complex behaviors? Here we have left-right, here we have forward, but how computationally intensive will this be, and how are you thinking about that?
This is computationally quite intensive, and that's part of this, part of the thing that I didn't mention. I put this on a supercomputer in Japan, world's 7th fastest supercomputer, ABCI. I scaled it up for that, and also used it, I also scaled up, I improved so this is the supercomputer we use. And then also I improved the algorithm, so it's like 6,266 times faster. So for example, if you want to do a mouse brain with 76 million neurons, originally it took 1,400 years. Now we could actually do it in 2 months, 26 days of calculation. So it's possible to do. And also develop a new type of hardware specialized for this type of calculation. We generated in collaboration with NEC something that has an old Cray supercomputer with a GPU and a computer and a CPU, and we generate a new type of supercomputer with new architecture that doesn't exist yet, other than this one, to solve this problem.
Great. Terry has a question. Terry, are you unmuted?
Yes. This is really exciting. This is exactly what we had hoped would happen with the Brain Initiative by generating these large data sets and letting other people have access to them. So this is very exciting. So here's my question. So it's a little counterintuitive. One might have expected that by increasing the dimensionality of the input, that you would be able to have a better and better model of what behaviors are. But what you seem to have shown is that by reducing the dimensionality, you're doing better. So do you understand that theoretically, or is there some explanation you can give us?
Yes. So it seems that if you actually increase the dimensionality too much, basically you have too sparse of a model. And also there's some gating activity that you actually have, that if you have all the information present, you just get an overfitted system, and you just get into these infinite loops. Basically is a problem of overfitting. But I think it's also the problem is like there is some property by having it basically nodes in the network that gates the information. And also this gating that you actually have on the low dimensional manifolds throws out some information that is irrelevant to move this thing forward.
So you're telling me that less is more?
Yes, sometimes less is more. It's like you don't want to have everything. Like if you instantaneously had everything communicating, you would not be able to calculate in a way.
Let me throw you one last question, Gerald, because the time is getting short here. Am I correct in thinking that this approach assumes the stability of the network over time, or can it handle plasticity as well?
No, okay. In a way, it's like right now the implementation is like it can do only it only has the network is basically the relationships are whatever the relationships are in the brain at this time, but it's like I'm actually developing a way to actually make it learn new behaviors from this, so it can become a learning algorithm. But right now it can adapt, and the network is basically a dynamic network. It's not a static network.
Great, great. Well, we have to leave it there, but thank you very much. Let's have a round of whatever kind of applause we can muster here online. And let me turn to our next speaker, Amy Prabhati from Biological Sciences and the Salk Institute. And Amy's going to get in here and share her screen when Gerald gets off there. Thanks a lot, Gerald. And she's going to talk about a new way of monitoring temporal information flow in C. elegans. A beautiful story here. So Amy, please take it away.
Hi, yes. Thanks everyone for coming to my talk this morning. I'm a graduate student in the Chalasani lab at the Salk Institute. And today I'm going to talk to you about my project using Campari in C. elegans and how I hope to use it to monitor temporal information flow during complex behavioral experiments. So the main model organism of choice in the Chalasani lab is C. elegans. C. elegans is a convenient model for the neuroscientist because all of its 302 neurons and their connections have been mapped, and most are genetically accessible through cell-specific promoters. It's also clear, meaning that fluorescence can be imaged in a live animal. This is a C. elegans adult with all its neurons expressing a fluorescent protein. There's a dense cluster of neurons near the head of the worm, which we loosely refer to as its brain. And this contains most of the worm's sensory neurons and interneurons. So one of my overall goals is to identify shared neural circuits in similar complex behaviors. C. elegans navigates the world mostly through its sense of smell and are able to remember positive and negative associations to specific odors. They then can modify their behavior depending on past experience, either moving toward or away from that odor. For example, C. elegans can negatively associate odor with starvation or with toxicity of food. I'm interested in how this behavioral plasticity can arise. My next and related goal is to record and compare neural activity over multiple periods of time during different epochs of these behaviors. For example, I could compare